Hello everyone, this is Nan. Today I'll be talking about the clock and wavefront model of segmentation, so please enjoy my video. One of the most fascinating features of the embryonic development of vertebrates is the complex yet highly organized segmentation along the anterior-posterior axes. As the posterior end of the embryo elongates, strictly defined somite chunks form along the central axes, rhythmic with time. Somites derive from the paraxial mesoderm, which are two strips of tissue sitting bilaterally to the posterior portion of the neural tube, the structure that develops into the spinal cord. Each somite is assigned a different structural fate through selective activation of a series of genes called Hox genes, and eventually develops into skeletal, dermal, or muscular tissues corresponding to their anterior-posterior positions. So, how does the somite segmentation achieve precise timely control that divides somite into such uniform chunks? And what determines which Hox genes to be expressed? A long-standing theory, the clock and wavefront model, illustrates a possible mechanism. The clock component comes from the periodically cycling genes, which counts the cycle of single somite formations. The oscillation could be observed here with the fluorescence. The oscillation of clock genes brings waves of chemical cascades, and somites arise exactly around the wavefront in the presomatic mesoderm. Let's take a look at how the whole mechanism works. Here's a schematic of the posterior end of the paraxial mesoderm. Naturally, there exists a chemical gradient of fibroblast growth factor 8, or FGF8, a protein secreted by the posterior end. On the other hand, already formed somite segments secrete retinoic acid, which creates another top-down chemical gradient. The two molecules are mutually antagonistic, meaning that they'll suppress each other's activity when come to a meeting point. Therefore, at the junction of the two chemical gradients, which is also referred to as the determination wavefront, the FGF8 level is brought toward a threshold that allows the clock cycles to happen. Let's take a look at how the clock works. FGF8 expression triggers Wnt3a, a signaling protein of the Wnt family. Wnt3a then activates two different pathways, triggering Notch and Axin expression. Notch then goes on to activate HERI1 and MESP2 pathways, while Axin serves as a negative regulator that returns to neutralize Wnt3a. Once the MESP2 is activated, it causes the expression of L-fringe, and L-fringe in turn targets back and inhibits Notch. Back to the main circuit, when HERI1 is expressed, it activates Ephrin A4, which is a membrane-anchored protein and is responsible of the actual segmentation. Hence, the rhythmic clock is essentially a result of feedback loops that constantly turns on and off gene expressions. Now let's take a look at how the clock and wavefront model puts together. Here are two cells located at different positions anterior-posteriorly along the paraxial mesoderm. As the posterior end elongates, the wavefront gradually proceeds posteriorly, and the FGF8 gradient leaves the anterior cell. We can notice that the identity of the two cells starts to differ, because the posterior cell goes through more gene expression cycles than its anterior counterpart. This tells us that the different time windows that cells sit in the gradient is a crucial factor in somite identity segmentation.